My talk will be about actually two topics. One is psychosis and the other one is about substance use. And actually I try to combine these two topics. So just a brief word about my background. So I'm a clinical psychiatrist interested in applying neuroimaging methods. And I'm particularly interested in the early phases of psychosis. So I'm not an expert in substance use, but obviously as a clinician and as a researcher we are dealing with these kind of confounding effects or confounding influences. So my talk will be about psychosis. As I said, I will introduce the early psychosis model and the concept, speak a little bit about cognitive effects during the early stages of psychosis, and then show imaging findings in early psychosis and the putative effect of substance use or misuse, and I use the example of cannabis use. And at the end of my talk I will give you an impression about the next steps and future directions of the field. So I'm sure you're aware of the staging model of psychosis. So as clinical psychiatrists we are usually dealing with patients in the stage 4 or stage 3 first episodes of psychosis or chronic stages, but we are aware that there is a kind of prodromal phase of psychosis, a pre-symptomatic phase. I will show you a few descriptions a little bit later. But you have to understand that before the actual breakdown to a psychotic episode, there is a phase of 3 to 5 years with the prodromal symptoms. And these are accompanied by different brain abnormalities. So brain structural processes or aberrant brain structural processes start much earlier. So if you think of this model of psychosis, as psychiatrists we are dealing with these patients with an established psychosis, with a certain severity of symptoms. But there is a group called clinical high-risk subjects or prodromal subjects or at-risk mental state subjects, so all the same more or less. And these patients, they have either so-called attenuated psychotic symptoms, so symptoms that are not as severe as in a psychotic episode. So for example, these patients have hallucinations but not as often as in a established episode. Or these patients have brief, limited, intermittent psychotic symptoms, which means they have psychotic symptoms for a short time and they disappear without any treatment. So and in the next slides I will go back to this ultra high risk group of patients for developing a psychotic episode. So what is interesting to study this specific group? As you can imagine, this is a group of people in their adolescence, 15 to 25, 28 years old, and they have not been suffered from kind of side effects of treatment. So they haven't been hospitalized, they haven't had any medication before, and their substance abuse is relatively lower compared to patients in their older age. So if we speak about ultra high risk subjects, you have to be aware that this is not the risk is not as high as you might imagine. So the overall risk to develop a psychotic episode is 35% of those who are considered as ultra high risk will develop a psychotic episode within the next 3 years. So you can decide for yourself what does it mean. So from a clinical point of perspective, we consider these patients as ultra-high risk for developing of psychosis, but actually only 1/3 will develop a psychotic episode. Now I show you some findings about cognitive effects. So these people do not only suffer from clinical symptoms, psychopathology in terms of positive psychotic symptoms, they also have cognitive deficits. And if you look at the comparison between those ultra-high-risk patients versus controls, you see an overall deficit in different domains of cognition. In particular, verbal memory, working memory is reduced compared to controls. But then if you look at those high-risk patients who really later develop psychosis, so the 35%, they have even more a decline in cognitive functions. So we reviewed this in a meta-analysis, and the meta-analysis was actually interesting, but there were some limitations. We were able to correct for age and publication years, antipsychotics, and so on, but we were actually not able to correct for substance abuse. So you might think that those who later developed psychosis are those who actually consumed more substances, and this is a correlate of cognitive effects. But we were not sure about that and couldn't address it in this meta-analysis. But there was, a few years later, there was a study looked at substance abuse in clinical high-risk subjects, and they compared those clinical high-risk patients with controls, with healthy controls, and they showed that indeed there is a higher use of cannabis and tobacco compared to controls, and this pattern of higher substance use continues over at least 1 year. But there was no difference at baseline regarding later transition, meaning that those who consumed more cannabis did not have a higher likelihood to develop a psychotic episode. This was published in 2015. So a few more words about cannabis. I'm not aware how many people consume cannabis here in the normal population, at least in Switzerland. There is it's more, I would say, it's more regular to consume cannabis than not consuming cannabis in a certain age group. And in this specific group of adolescents, young adults, as I said, it's more common to use that. So why are we so interested in cannabis? So cannabinoid receptors are widely spread all over the brain, but in particular they are distributed in the areas of the anterior cingulate cortex, prefrontal cortex, and temporal cortex. And these areas of the brain are exactly the ones which are disturbed or altered in psychosis. So it's not surprising that cannabinoids in general do not only lead to cognitive deficits, they are also able to induce psychotic symptoms. So let me now show you a few imaging findings in the early stages of psychosis. So why do we do imaging at all in psychiatry, apart from the clinical indications to exclude some disorders, brain tumors, and so on? In psychiatric research, we are interested because imaging may bridge the gap between the clinical syndromes we are dealing as clinicians and the genetic basis for disorders or for these syndromes. For instance, if you look at specific genes as COMT, these genes lead to a disruption of brain activity or brain regions, and these disruptions lead to certain syndromes as working memory deficits. So that's the overall idea to use imaging methods. So one study that's been published in 2008 deals not with early psychosis patients, but with established psychosis patients, in particular schizophrenia patients. And this group around René Kahn, they showed that patients using cannabis, schizophrenia patients using cannabis, have a reduced gray matter volume compared to those to controls, that's clear, but also reduced compared to patients not using cannabis. And this corresponds nicely to the increased ventricular volume. I'm sure you're aware that the most replicated imaging findings in psychiatry is the increased ventricular volume in schizophrenia. And again, so the reduction of gray matter corresponds to an increase of ventricular volume in patients using cannabis compared to the other ones. So this indicates that cannabis use is associated, at least, maybe leading or even strengthen the gray matter reduction, which is already present in schizophrenia. Review, which I can really recommend to read if you're interested in this topic, has been published 2017, and this review nicely summarizes the results of patients with schizophrenia spectrum disorder compared to schizophrenia spectrum disorder with substance abuse. And they showed that if you have a combination, so a comorbid, if you want, a comorbid substance abuse in schizophrenia patients, that these patients have more severe brain structural alterations. And they tried to look at differences, or they wondered whether this occur immediately after the start of the disease or not, and they found that the brain structural alterations are prominent in the substance use or comorbid substance use group only after 5 years of illness or longer. So that's interesting, but I will show you a few more other findings, but I can recommend this paper to read. So coming back to the early phase patients, you remember the ultra-high risk patients? So the first study published on this group was published in 2003, and the authors showed that over the transition from an at-risk mental state to a first episode psychosis, there was not only a cognitive decline and more symptoms, there were also a reduction in gray matter volume, and we have replicated this finding in a different cohort. But the question whether this is related to substances was not clear. So we did a analysis in our ultra-high risk patients and first episode patients. So you see, so you see that the sample size is not impressive. So Basel is a relatively small town, so only a few patients, but I will come back and show you later the directions for future research. And when we now looked at the cannabis users and the non-cannabis users, we could see a negative effect on cannabis use on the volume, on brain volumes, and in particular of the cingulate volume. So the higher the dosage of cannabis, the lower the gray matter volume in the cingulate. And again, this is interesting if you think of the distribution of cannabinoid 1 receptors. So both at-risk mental state patients or clinical high-risk patients and first episode patients seem to be particularly sensitive to exogenous activation of these receptors. So, but you may ask what mechanisms are underlying these interactions between cannabis use and transition to psychosis. And there are a few hypotheses. Hypothesis about neurogenesis, neurodevelopmental processes, but I think the most interesting one is the interaction between genetic factors, so the underlying vulnerability to psychosis, and environmental factors as trauma and as cannabis use. And there is probably an interaction between both. Interesting to discuss, maybe in the later discussion we can go into detail, is that now I've spoken about cannabis use, but actually cannabis use is a very broad term for using 60 or even more phytocannabinoids. And you know that THC, as the main psychotropic constituent within cannabis, is the one that leads to psychotic symptoms. But there are other substances as cannabidiol, which has been shown to have, on one hand, anxiolytic effects, and on the other hand, antipsychotic effects. And so it's interesting to discuss whether we should actually speak about THC instead of cannabis use. I think in March last year, there was a study showing that CBD as an add-on to regular antipsychotic treatment can improve the treatment of schizophrenia. But to make the story even more complex, there were a few more studies, and they looked not only on cannabis use, they also looked at other substances. So, but again, they compared clinical high-risk subjects, users versus non-users, and looked at certain brain regions, thalamus, hippocampus, amygdala. And what they found was, when you look at the three regions, at the first glance, again, there were differences between users and non-users. But then, if you correct for use of alcohol and tobacco, it wasn't so clear. So if you correct for these substances, then the results for the thalamus and amygdala basically disappeared. There was no difference anymore. And only the hippocampus was different. But again, that actually confirms our hypothesis, because cannabinoid 1 receptors are widely distributed in the mediotemporal cortex and therefore in the hippocampi. So to sum up, cannabis use, or actually THC use probably, in psychosis is associated with an even more increased volume loss of global brain volume and specific brain structures as a cingulate cortex hippocampus. And psychosis patients, but in particular early psychosis patients, may be particularly vulnerable to further brain volume loss. But you have to have in mind that other factors as alcohol use and tobacco use are highly interrelated with the cannabis use, so you have to control for that. So a few more words about the next steps, about the research directions for the next few years. So there are still open questions. So are the brain abnormalities only the consequence of the substance use? Probably not, because there are also there independent of substance use. We have to deal with the THC CBD story, so different differential effects within the cannabis. And we have to increase the sample sizes for these kind of studies. And there are large ongoing studies now. I will show you a slide in a few moments. And this is the most interesting development in the last years. I think there is a movement away from these kind of group imaging studies I've shown you so far. So I always showed you a group of patients with psychosis, group of patients with clinical high risk versus group of healthy controls. That's interesting in a way to understand the basis, but actually it's not worth and not helpful for clinical decision making. So there is a trend towards individualized imaging. And I'm sure you're aware of these developments in the pattern recognition field. Google, Facebook, everyone is using that basically to find patterns in certain data. Not just neuroimaging data, you can use it for all kinds of data, but you can use this technique for imaging data and as well, and you can do classifications or even predictions using these kind of individualized imaging. And in a larger cohort of people, we were able to predict the outcome of patients, functional outcome, on the basis of gray matter volume, just on 10 minutes structural MRI scan. One was able to predict later functional outcomes. And you can imagine that you can nicely combine this with substance abuse data if you would include that. So I have to come to an end, but I would like to recommend very nice trans-diagnostic risk calculators. These are very rough now, but so far only easy demographic variables are included, but if you add cannabis consume or tobacco or alcohol consume, you might even increase individualized risk calculation prediction. So colleagues here from Milan, Paolo Brambilla, are also working together in large-scale projects, European projects dealing with these topics.